trapped under the water for about an hour until they were able to rescue her out. The really astonishing part is once she was kind of re-emerged, they pronounced her dead on the scene, but they ended up taking her to the hospital anyways. And after about 9 hours of resuscitation and several months of rehabilitation, she made a full recovery. And at the time, and actually this is a record that still stands, her body temperature was the lowest that was ever recorded in an adult. It was lower than 14 degrees Celsius. And the fact that she survived and actually went back to skiing in the last couple of decades was really remarkable. And what it really emphasized to us was kind of the importance of brain temperature and how sensitive it is to temperature. So we've known this for a long time. We've known this especially with body temperature. So the idea that we can cool people down to provide some protection is actually not new. It's been done for many decades. Often it's been done after things like cardiac arrest, where they would literally used to take bathtubs or even small children's pools, fill them with ice and put people in them. And they found that that cooling, especially after something like cardiac arrest or stroke, actually provided some neuroprotection, which was really amazing. And then the reverse, of course, is also true, that higher temperature after stroke or after a lot of conditions or any sort of fever actually is often related to worse outcomes. So this is a plot of outcomes after stroke on the stroke scale, and you can see that this hyperthermic case always has the worst outcomes relative to the normothermic or even hypothermic case. So we know temperature is really important, but really brain temperature has been relatively understudied over the years. So from a very basic thermal perspective, the way that we know that brain temperature is regulated is through cooler arterial blood that comes from the heart. And we know the brain is a very metabolically active organ, so it generates a lot of heat just from cerebral metabolism. And this arterial blood ends up acting as a heat sink, taking all of that heat that's generated by the tissue and taking it back through warmer venous blood back to the heart. And so that's really kind of the principle that really drives a lot of the work that we do, which is kind of understanding this thermoregulation in both health and disease. Now, a lot of the early brain thermometry work has been really challenging, but it was really propelled by a couple of important inventions, one of which was the thermocouple, which of course is just basically a temperature probe, and the other is the stereotactic device. And so these were really the tools that allowed us to start studying this in mammals. And some of the first brain thermometry work in mammals was actually done in a feline model. I think this was the first reported measurement of direct brain temperature in a mammalian model. And so on the bottom here, there's this plot right here in A. This is temperature as you're moving the probe from left to right. So you can see that the midbrain is higher than the periphery. And then on the top here is moving that same temperature, that thermocouple, from the skull down to the deep brain. And you can see, again, the deep brain is warmer than the skull. And this makes sense based on conventional wisdom. But of course, the challenge of this method is that you have to, for example, in humans, you have to open the skull, do a craniotomy, and then insert the probe. So you can imagine that's not an extremely popular technique. And it really comes back to this kind of obscure physical chemistry principle known as Heisenberg's uncertainty principle. And let's try to move this. Sorry. Well, it's okay. No, it's okay. So the Heisenberg's uncertainty principle is very simple. Basically, you cannot know the position and momentum of a system exactly, right? There's some uncertainty, and that's constrained by Planck's constant. But it really applies in this case, and in imaging very broadly, to the fact that every time you make a measurement of a system, every time we image a system or probe the system, you actually disrupt the system a little bit itself, which means what you're measuring is the perturbed system. So if we think about that in a very simple context in terms of brain temperature, what we want to do is we want to try to minimize all those perturbations to the system while still getting the same information. So our brain thermometry tools really can kind of span this spectrum. So on the very high end of perturbation is sticking a thermocouple into somebody's brain. Of course, that's a very high degree of perturbation. A little bit less so is imaging, something like MRI. And I'm going to talk a little bit about how we can measure temperature using MRI. And then on the farther end of the spectrum is modeling. So can we create a model where we have some input data? We don't necessarily have to perturb the system more than we would clinically. And can we get brain temperature information using this minimally perturbed system? And so that's really what I'm going to focus on today, is how we can kind of span this spectrum. So with MRI, there's a really nice principle that allows us to measure brain temperature using an MRI scanner. The fact that there's a built-in spectrometer in every clinical MRI scanner helps us out in this case. And water, of course, is very temperature sensitive. We know this. And what we can do is we can look at the chemical shift, or basically the response of water in a magnetic field to temperature relative to something that's not temperature sensitive. So something like NAA, N-acetyl aspartate. And so what happens as the temperature increases, the water hydrogen bonding around the protons basically breaks, increases the electron shielding. And what that does is it basically protects those water protons. And the water peak can shift to a lower frequency. So you get this upfield chemical shift. And this difference between water, which is temperature sensitive, and some other molecule, such as NAA, which is not, turns out to be very linearly correlated. So this has been shown many times before. We've done this in some early work, just doing this in a phantom, a temperature-controlled phantom with water and NAA. So we wanted to do this basically in an MRI scanner so we could demonstrate that we could do this in the same system that we would eventually put our patients. And you can see that that chemical shift difference is extremely highly correlated to temperature. About 0.01 degrees ppm per degree Celsius is a pretty classic relationship between these two. So we can use that principle to get quantitative brain thermometry from MRI, and we can actually get this in an absolute way. So we can use this calibration to say, for example, that region of the brain is 37.5 degrees Celsius, so it's not a relative measurement. So we've used this MR technique in actually a number of applications. I want to talk a little bit about this. So this is all experimental thermometry work. So one of the things we wanted to do is just really validate the measurement and understand how much fluctuation we got. So in this particular experiment, we looked at repeated measurements within 2 minutes, and we did that twice. And those measurements were just to say, okay, in 2 minutes, the brain temperature of a human probably won't fluctuate drastically. And so we would expect the temperature to remain constant. And that's what we found. We saw no significant changes in this very short time period. But then we allowed the patients to be in the scanner, come out of the scanner, move around within about a 60 to 80 minute time period. And we wanted to say, on this very short time scale, do we actually see any temperature fluctuations? And I think one of the motivations for this work was, we know in this idea of resting state, we know that there's a lot of fluctuations that happen just in the resting brain in terms of blood flow and metabolism. And so we actually saw the same thing happening with temperature. We actually saw a significant decrease in temperature just over a very short time scan. And the patients were largely resting during this time. They would get up for a few minutes and walk around, but for the most part, they were resting. And so we really took this as evidence that even in the fairly rested brain, we actually can get some fluctuations on very, very short time scales. So people have seen this over weeks and months, of course, but on an hour's time scale. This was kind of the first evidence that there is some fluctuation over time. We've also looked at this in a number of different cohorts. We looked at this in healthy adult athletes. So these were young adults. And we looked at them over the series of a season of college basketball. And these were healthy adolescents. So they were people who never got a concussion or a traumatic brain injury. They played a season of basketball. So there were some kind of mild injuries just from playing college sports, but nothing serious, no concussions, no loss of consciousness. And we saw in this healthy cohort that we have a very consistent increase in temperature in the middle of the season. So we scanned them pre-season, mid-season, and post-season. And in many brain regions, we see this kind of increase in mid-season, which could be due to a number of different things. We actually don't think it's due to inflammation. We measured peripheral inflammatory markers and didn't see any changes. So we think it could just be due to higher brain activity over the course of seasons and due to athletic conditioning. Yes.
So this is just like one session measurements, or these are like many, like when you say mid, is it just really 1 point for each subject?
That's right. Yeah, 1 point. So before the season started, before they played any games, right in the middle of their collegiate season, and then postseason after the postseason ended, even with some kind of playoff games. Yeah.
And mid was defined just based on temporally, or just like how many games they've done? Or I'm just curious.
Yeah, it was basically kind of, their season was about 4 months. So it was at the end of the second month. Yeah, yeah. It was a little bit correlated with when their winter break was, because they're collegiate athletes. And so some of it was practical, but it was basically temporally right in the middle of the season.
Right.
Yeah. So we've moved on to looking at this in kind of more extreme models. And a lot of the work we've done in the lab is focusing on stroke or cerebrovascular disease. And the reason for that is we think that's a very nice model where temperature is likely to change because you have a very, large change in blood flow. And remember, blood flow is really what allows the brain to be cooled. And so we think stroke is actually a nice model for where brain temperature might be a nice imaging biomarker. So we did this in three primates. So when we started, they were healthy, and then we had an induced ischemic stroke. And then we monitored them for several hours the week before, and then immediately after the induced stroke for several hours. And so these images, the cooler temperatures are lower, the warmer, the red temperatures are higher temperature. And you can see the final diffusion weighted image showing the region of the infarct. So it's a fairly large induced stroke. So what we found in this case was for the healthy primates, monitoring them over several hours, their brain temperature and their rectal temperature or their body temperature were very tightly coupled. We saw this, even if we saw fluctuations in one of the primates, we saw that that coupling was consistent in the healthy primate brain. And then post-ischemia, one of the things that we found, which is actually consistent what has been shown with patients, is that the brain temperature actually increases at a much higher rate than the body temperature. So there's this decoupling that happens after injury, especially after this extreme injury. And we actually think that that decoupling is a very important marker of health after something like a stroke. So we think that that's going to be really important in future studies with patients. And then we have looked at this also with patients who have chronic cerebrovascular disease, primarily some type of stenosis or narrowing of some arterial vessel in their brain. And we did a pre-post study where we acquire this kind of multivoxel thermometry. And then we give them a drug known as Diamox or acetazolamide, and it's a vasodilator. So it effectively widens their vessels. And so we actually do this clinically now. And then we take thermometry again. And the idea is that in a healthy individual, if you dilate their vessels, they're going to have an increase in blood flow. Their metabolism should stay the same. So you should get cooling from doing this. And actually, we see that with other markers such as cerebrovascular reserve. We see this kind of dilation in healthy regions and no change in impaired regions. And so what we see here in this plot is we see in some of these regions, we actually see no change after giving the vasodilator. So this brain thermal response is just a normalized difference pre-post. And so those regions that have no change and shown in red, that's a very impaired region. Actually, that correlated very strongly with our cerebrovascular reserve measurements. And you see some areas where we would see what we expect, which is that cooling after having a vasodilator. But this was an individual with very chronic bilateral disease. And one of the really important things about this is this DWI image was negative. So we don't see any lesions on this image, but we do see this very, in some ways, high impairment of cerebrovascular activity. And so we think that, again, this is a nice marker that will complement what we're already doing in stroke and in cerebrovascular disease patients, because even if we have a negative DWI, we can still see some texture in these images. Yes.
So on the image on the left, what was your resolution for your actual temperature mapping? Because you have kind of some super pixels there, but you clearly have detail in those pixels. So
yeah, I'm gonna say it was about 1 centimeter isotropic voxels. So it's fairly large because we're actually using chemical shift thermometry, so it's spectroscopy. Yeah. And some of the detail is let's do two things. One, interpolation, and two, it's actually a transparent color map overlaid onto the image. So some of the texture that you see is that T1 weighted image below. Yeah.
got it.
Yeah.
But those white squares do represent what
the spatial the nominal spatial resolution. Yeah. Yeah. That's a great question. Thank you so much. So again, as we're kind of looking at this kind of degrees of perturbing our system, a lot of the MR thermometry, it's not an overly perturbed system, but it is a significant burden, right? To do this MR thermometry takes some time, and we're adding that onto their clinical scans already. So one of the things we wanted to do is say, okay, can we take this and can we do this in a less perturbed way? So instead of doing this very long chemical shift protocol, can we model the brain temperature just from structure? And that's really what we've spent a good amount of time doing. So in terms of how we model the brain, so we start from a very simple system. We have 3 domains. We have arteries, which again are bringing that cooler blood. We have porous tissue, which is tissue and capillaries. and we have veins. So those are the domains that we model heat transfer through. And then we look at 3 different heat transfer modes. So we look at heat transfer via advection, which is due to, moving fluid causes advection. We also look at conduction, which are 2 solids touching each other, the heat transfer between solids. And then we look at convection, which is a mix between the 2. So I won't tell you every detail of these equations. I won't bore you on a Friday afternoon, but I will say it's a closed system, so they're all equal to 0. So within each domain, we conserve energy, mass, and momentum. But really importantly, what we were able to do is do this across domains and across over the whole brain. So we treat the brain as an adiabatic system, and by preserving this, we're actually able to demonstrate kind of for the first time that we can create a fully conserved model of human brain temperature, mass, momentum, and energy conservation within the tissue. So in terms of taking these models and doing something with them to predict brain temperature, what we wanted to do was take MRI data from each individual. So we wanted to create a personalized model. So we have a fully conserved model, so we know the model doesn't violate any principles of physics, but then we wanted to do this for each individual person. So the idea is that we're going to take their T1 image, their arteries, their veins from their MRI images. We're going to validate that with MR thermometry, and then we're going to use our heat transfer equations to model the brain temperature. And our thinking here was that most of the time, a lot of our patients are already going to get these images. So they're already going to get T1, and we can get their brain temperature without even having to do the thermometry. In this case, we, of course, wanted to validate that. And so we do some co-registration of all the images. We do some segmentation and a little bit of vessel augmentation, because, of course, what we get from MRI is fairly crude. And then using the tissue and the arterial and venous structure, we can put that into our bioheat equations and model that across the whole brain. So what this looks like here is if we have this MRI data from each individual from our T1 or from our structural imaging, we can create these metabolic heat maps. And again, metabolism is heat generating in our model. We can take our arterial structure, which is shown in red, our venous structure shown in blue, and we can predict cerebral blood flow. So we can create these cerebral blood flow maps. And then along with our bioheat equations, we can create these temperature maps. So we can create these 3D predictions of individualized human temperature across the whole brain. And of course, we wanted to validate this with our experimental MR thermometry to see how close we are. Of course, both have pros and cons. But we use this threshold of 0.8 degrees Celsius because that's some of our uncertainty in our MR measurements. And we can see in most regions of the brain, we get a very tight agreement between our modeled and our measures across the hemispheres. We have a couple regions which are a little more challenging. The cerebellum is mostly because we don't always acquire that in our experimental thermometry due to how we acquire the whole brain data. So there is some more variation there. But for the most part, we get very tight agreement between our model and our measurement. So we wanted to apply this, and again, stroke is really our primary application for most of this. And the idea with stroke, and the reason why it's such a good model, is if you have a blockage in an artery, like you would have in an ischemic stroke, the center of that would be kind of your infarct region, and that would be your irreversible brain damage region. And then right around that, you're going to have an area where there's going to be reduced blood flow because you have that infarct or that ischemia, but you're actually going to still have active metabolism. And so that region around the infarct is known as the penumbra. That's really often referred to as kind of a potentially reversible region. But from a thermal perspective, it's going to have higher temperature than the infarct because the infarct won't have any metabolism anymore because that tissue is dead. So now you have this contrast from a thermal perspective where the center of your infarct is cooler than the reversible region outside of it, or the penumbra. And so this is why we think stroke is such a good model, because naturally there is going to be this difference in temperature. So we've modeled this in a couple of different ways. This was actually an angiography image from a patient with a right MCA occlusion shown in yellow. And so we took their vessel structure and we input it into our model. And we predicted what the temperature evolution would be over time. And what we found, again, was exactly what we expected. The penumbra had the highest temperature, the infarct following that, and then the healthy region had the lower temperature compared to the two. So we think there is some kind of thermal insulation in the infarct that makes it higher than the healthy brain. But again, we do see that separation between the penumbra and the infarct. And people have done this experimentally in animal models, and they see these very similar patterns. We also wanted to look at this from a treatment stratification point of view. So modern treatment for ischemic stroke is really first upon admission, give some pharmaceutical, typically it's TPA, and this dissolves the stroke, that's standard of care. And then there has to be a critical decision that's made after this TPA, which is to do endovascular thrombectomy or mechanical removal of the clot. And thrombectomy has really changed stroke management, across the country. Because it's been very effective, but it's very challenging because the mechanical removal is very invasive, of course. And so it can be very dangerous if someone doesn't have enough recoverable tissue to do that. And then in some cases, the infarct is so large, it's also not beneficial for the patients. So there's this very kind of clear decision point that has to be made. And normally what's done is to use CT perfusion to do this, and that's been fairly effective, but there's still a number of cases where the patients have poor outcomes. So I think temperature might be a nice complement to what's already being done in the clinic. So I want to give you just one example of this. So the classic selection, of course, this varies. I feel like there's a new guideline for thrombectomy every year, but the current guideline is the ischemic tissue over the infarct, that ratio has to be greater than 1.8. So there has to be enough recoverable or salvageable tissue to make it worth it. And then the infarct volume has to be less than 70. And of course, they have shown that, larger infarcts can still benefit. But the idea is that you have enough salvageable tissue and that your infarct is not so large that it will benefit the patient. So that's the standard clinical guidelines. So we had a 58-year-old male, had thrombectomy after an acute ischemic stroke, but the patient did not have reperfusion after this thrombectomy. So even when they went in and removed the clot, they actually didn't get restored blood flow. And so their conventional ratio of 5 and 45 stratified them for thrombectomy, which is why they underwent that procedure. And we did the same thing retrospectively with temperature, where we took their ipsilateral and contralateral temperatures. We used standard deviation thresholds to identify ischemic and salvageable tissues. And we found that if we used temperature calculated ratios of those regions, we would have found this patient would have had a lower ratio of salvageable tissue and a larger infarct. They actually would have been excluded. So we've seen this in a number of cases. We've just done this in about 4 patients so far. We've seen that temperature is actually very consistent with what we see in the clinic in terms of who responds and who doesn't. So we certainly are never going to replace CT perfusion in this, but we do think this is a complementary metric, especially for stratifying patients for this very invasive treatment. The other application where we think that temperature, of course, is going to be really important is in these types of cooling treatments. So we know that the brain responds very well, especially if it's cooled after an injury. It can provide a lot of neuroprotection. There's actually a lot of treatments out there that are kind of positioned to cool the brain very quickly after these injuries. And so there's basically 2 ways to cool people. One is to put a cold helmet on their head. Let me just play this video for you. So put a cold helmet on their head and basically reduce the temperature, the external temperature, by about 10 degrees. So we've modeled this. If we take our brain temperature map, we reduce the temperature to about 10 degrees Celsius, which is equivalent to putting ice basically around their head in a helmet. And we see that the external part of the brain, the skull, is cooled very efficiently, but the inside of the brain, or the brain tissue, really isn't cooled even over quite a bit of time. The other way that we can cool people is by cooling the arterial blood. So if we remember that arterial blood comes in from the heart and actually acts as a heat sink. And so we can do this in a number of ways. We can do this by injecting cold saline into a major artery. We can also do this by putting heat pads or cooling pads on their major arteries. And you can see just in this image, just by doing that, decreasing the inlet temperature, the arterial temperature, by 0.5 degree Celsius, we can cool the brain much more efficiently. And so there's a lot of thought that a lot of the reason why some of these cooling therapies are not effective is because they're actually not cooling the deep brain. And of course, we're not measuring brain temperature ever in the clinic, especially not in neurosurgical cases. So we think this type of modeling and kind of prediction can really help us guide, which of these therapies are effective and why some people are responding and some people aren't. So in terms of taking this model kind of to the next step, the momentum conservation was really the key thing that we've finally overcome. But I think one of the major things that we need to improve is really the vessel structure. And so that's the challenging part. We're just getting that from fairly crude angiography or even post T1 contrast phonography. But we recently got the first 7 Tesla MRI scanner in the state of Georgia. And so we're trying to really use a lot of those capabilities. I don't think I have to convince this audience of why that's important, but you can see just from just a non-contrast angiography image, you can get a lot more signal. And one of the things we hope to do is be able to map those blood vessels much more accurately. Because really in our model, it's the diameters and the lengths and the paths of those blood vessels that really determines a lot of the temperature modeling. So I want to just summarize this temperature part, and then I want to give you a brief overview of some of our other projects. But really, kind of the take-home message, especially from a clinical point of view, is that higher body and especially brain temperatures lead to worse outcomes in patients. I think thermal modeling using data that we would already collect in the clinic is a much lower burden alternative to doing thermometry. It's definitely a lower perturbation than implanted probes in the patient. So we think this is one avenue for this, because a lot of the patients are already getting an MRI scan. So now we can use that data to create these temperature maps. And then from a very big picture perspective, I think there's a lot of considerations of kind of moving to high field. There's also been a big push in the field recently to move to lower MRI fields. And if we think about this just kind of from a climate change and energy point of view, there's kind of two sides to this, right? High field scanners, of course, require a very large energy demand, and hospitals are one of the highest energy users in the country. And it's really second only to data systems, which these very high computational demanding systems, such as what's often used with low field images to kind of reconstruct. So I think there's kind of a paradox here in terms of where we're going in the future. I don't have an answer to that, of course, but I think it's good food for thought. So as Christine mentioned, a lot of the work we do is focused on MR spectroscopy. So we really have a kind of fully translational pipeline where we develop methods and then we want to kind of see them through to the clinic. And I want to just give you a couple of examples of that. So one of the areas that my group has been working on for a number of years is kind of MR spectroscopy in the realm of brain cancer. And one of the things I want to highlight, particularly in cancer, is I think there's a very big space for metabolic imaging. I think we know that. And I think there's a lot of methods that are already being used clinically, things like PET. We recently published this review where we really highlighted kind of the road to metabolic imaging in cancer. I think that's a really big clinical need. And as we were talking about at the workshop today, MR spectroscopy is actually currently being built. There's a CPT code for it, and something that people don't always know, and we're not using very much. The place that we're really using it for is in tumors. But actually, there has been a pretty good trend of reimbursement, both by Medicare, Medicaid, and also private insurers are paying sometimes up to $3,000 for a spectroscopy scan alone. And so kind of that change in coverage, especially at the national level, has been really a good thing for spectroscopy, especially in the cancer realm. We also look at traumatic brain injury. So we look at this from a number of different avenues, from very mild to very severe, where patients are in a coma after, for example, an automobile accident. We do a lot of work in this space with athletes. So we just published a study looking at high school football players and the intervention of this, what we call a jugular vein compression collar. And it basically kind of reduces the brain slosh and can affect a lot of the metabolic metrics, as well as other kind of standard DTI metrics that you might look at in traumatic brain injury. So we think there's a lot of room, of course, for MR, but also MR spectroscopy in this space of traumatic brain injury. We've done a little bit of work in kind of an MRI adjacent space, which is looking at kind of the effects of the high cost of MRI scans and socioeconomic status on MRI and MRI outcomes. And so this is actually a map of kind of a socioeconomic status indicator known as the area deprivation index across the US. So red is lower, so it's more disadvantaged. Blue is more advantaged. And so we can look at this in terms of where our patients are coming from. And what we found was that we found a couple of things. So one, the higher out-of-pocket costs for MRI were directly correlated to longer follow-up times in patients with multiple sclerosis, where you have regular repeated MRI scans. So I suppose that's not surprising that the more expensive it is, the longer you wait to have one. And we also found that socioeconomic status of the patient directly correlated to the acuity of their brain scans when they came into the center. And we saw this over a very large number of patients. So these are kind of related but important topics if we're thinking about kind of translation of a lot of these methods to the clinic. Of course, these important factors such as financial toxicity, and socioeconomic status matters. And then lastly, one of the things that we have ongoing is a clinical trial, a phase 2 clinical trial looking at HIV in the central nervous system. So there are actually a lot of different kind of pools of HIV in the system of patients. And antiretroviral therapy can actually get rid of a lot of these reservoirs, especially in the periphery. We've shown it you can basically live without detected HIV in your bloodstream, which is really phenomenal advance in HIV science. But one of the things that happens is HIV will remain, even after years of being on ART, will remain and pooled in a reservoir in the central nervous system. And it leads to this really common, this very, very prevalent syndrome of people having neurological effects from HIV, so really severe cognitive effects. And so one of the things we're doing is giving this drug that can cross the blood-brain barrier. It's actually a drug that's already approved for rheumatoid arthritis, and it can clear this reservoir of HIV in the CNS. And one of the things we want to do as imagers is really understand the effect of that. So we set up a fairly standard imaging protocol at 7T to do this. And one of the things that we've done with this is to set up a 7T diffusion-weighted spectroscopy protocol. And I wanted to highlight that because it's available. So if anybody's interested in doing this, it took a little bit of optimization, but we'd be happy to share that protocol. And what's really nice is we can look at these kind of direct markers of inflammation using this protocol. And so that trial is ongoing. I think we've completed 10 patients and we have about 50 more. So hopefully next time I'll be able to give an update on that. All right, with that, I want to thank you. I wanted to highlight our center here, mostly because, as I think all of us are, we're always looking for good postdocs and research scientists. So if you're interested, please reach out. This is the city of Atlanta, and this is actually our new health sciences research building where our imaging center relocated, and then also my lab is currently. So with that, I want to thank you. I want to thank my team and our collaborators and our funding sources, and I'd be happy to take any more questions.